with 5 minutes to go. And our keynote speakers here. Our plenaries are here. And just to check before we start, is Corinne Yvonne here? Corinne Yvonne? Okay, so let me just check if she's gonna be online. So we'll be starting around 9 o'clock. I'll just come and do the wrap up with about 2 minutes to 9, and then we'll start the event. All right. Good morning, everyone, once again. I think I heard one person say good morning. Good morning, everyone, once again. Okay, at least I know that the room is full. Well, has people. So we're about to start day 2 of this 5-day conference. And as I mentioned yesterday, I believe yesterday was a good start to the conference. We started off with quite a show, which basically summarized our 5 years and had a very inspiring keynote from Emeritus Professor Ernest Aite, talking about the dearth of research in Africa and then the promise of what's to come. And we wrapped up with, I think, what I found were quite stimulating talks. And we hope that that was just the beginning and we're just going to keep going on till we hit the crescendo on Friday. So our first keynote of today is, he was one of our PhD, I believe when I started here at WACCBIP, he still hadn't defended his thesis. So he's one of our PhD products. And him along with Elvis, but primarily him, a lot of his work is what built the hearing impairment work that we've done here at WACCBIP. And he went on to do a postdoc at UCT, University of Cape Town, and has followed that up with another postdoc at the NIH. And when we thought of doing this, I thought it was only right that he be one of the keynote speakers because he's an accomplished scientist. And without much ado, I'll introduce Dr. Samuel Mauli Adade to give the first keynote of today.
Thank you very much, Dr. Kwashi, for that kind introduction. It's good to be home, where it all began. My journey into hearing loss research began here in WACCBIP, and I'm so much happy to be here today. Before I start, I want to use this opportunity to thank the faculty of WACCBIP, the WACCBIP machinery, and everyone who is involved in training students here for the training I received when I was here. And also to thank the organizers of this conference for a job well done. I want to share one fun fact before I go into the presentation. So during our comprehensive exam, before our comprehensive exam, we all wanted to maintain the high standard and quality of science that is practiced here in WACCBIP. So we're all working hard. I remember one night, for the first time, I took several bottles of one energy drink called Rush in Ghana. And fortunately, it was 3 days to the exam. The exam wasn't the next day. And for a whole 24 hours, I wasn't able to sleep. From that day, I've never taken Rush again. So over the past 8 years, I think 7 years, sorry. Over the past 7 years, I've been working on hearing loss with the overall aim of identifying candidate genes and also understanding the pathobiology of hearing and look for variants that are population-specific with the aim of developing population-specific diagnostics for hearing loss. So today I'll be giving you a talk on what I did during my PhD. So hearing loss has been on the rise for some time now. WHO estimates that about 1.5 million people are currently living with hearing loss. Then in 2050, about 10% of the world's population would have some form of hearing loss. That's the estimate from WHO. When you come back down to Ghana, from our population and housing census, about 1.7% of the population are having some form of hearing loss. So we consider it as the silent epidemic. When you look at hearing loss, you can classify it based on the part of the ear that is affected. When sound travels through the ear, it goes through the ear canal and hits the tympanic membrane, which is the eardrum, generates vibrations which are carried into the inner ear. In the inner ear, you have the vestibular piece, which is responsible for spatial orientation and also balancing. Then you also have the cochlea. The cochlea is responsible for hearing. So you can hear me now because of the cochlea. And we have a lot of genes that are expressed in the cochlea to help us hear. Now, when the inner ear is affected, it results in sensory neural hearing loss. And when the outer ear or the middle ear is affected, it results in conductive hearing loss. It's easy to, I won't say easy, it's much more easy to cure sensory neural hearing loss. Sorry, to cure conductive hearing loss. So now that we are all here, if you cover your ear with your hand, then you can feel what conductive hearing loss feels like, because you cannot hear me very well. And through surgical procedure, it can be corrected. We also classify hearing loss based on the threshold of sound that an individual can hear. And here you'd see a graph. It's called the audiogram. We use the audiogram to examine the threshold of hearing of an individual. On the vertical axis, you see the various hearing thresholds, and on the horizontal axis, you see the frequencies that are tested. And on this graph, I have images to help you appreciate the level of sound produced at each threshold. So based on this, we can classify hearing loss as normal, people having normal hearing, then hearing loss as mild, severe, moderate, or profound. So I'll be using these terms as we move on. And also permit me, I'll be interchanging hearing loss and hearing impairment. They are all the same. Hearing loss can be caused by environmental factors. Specifically, we can have sickness such as meningitis, responsible for hearing loss. We also have indiscriminate use of antibiotics, such as gentamicin, resulting in hearing loss. Accidents can also cause hearing loss, and especially in the aged population, sometimes noise pollution can cause you to lose some of your hair cells and have some level of hearing loss at some frequencies. There are genetic factors that are associated with hearing loss. We classify the genetics part of hearing loss into two. We have the syndromic hearing loss that's linked to known syndromes, and non-syndromic is not linked to any syndrome. With the genetics of hearing loss, the genes that are associated are well studied in other populations, but when you come to the African population, we really don't know what are the main drivers of hearing loss in the African population. Here, we try to put a map of the various genes that have been studied. And on this map, you see that a lot of the sub-Saharan African countries don't have any study on hearing loss. And the gene that is mostly studied across the whole world is GJB2. GJB2 codes for gap junction protein. And these gap junctions form junctions between cells and allow cell-to-cell communication and help us to hear. So the common variants in GJB2 are, when we map them on the African map, you see that they are mostly in the North African countries, and sub-Saharan Africa is almost empty. And there is a founder variant reported within the Ghanaian population, and this variant, I'll talk to you more about it as we move on. But this makes Ghana to be a good place to study this particular variant. A lot of the studies in Ghana are focused on the environmental causes of hearing loss, with only 2 studies that were on the genetics. The first one from a village called Adamurabe, which is known for high prevalence of hearing loss, and another one was a nationwide study in 2001. Then our study followed up in 2017. So we had a fallow period where no one was interested in the genetics of hearing loss. So we sought to identify or determine the environmental causes of hearing loss and also the genetic causes. Then design population-specific tools for screening this condition within the population. Our sampling strategy is such that we go to the schools for the deaf, identify patients who have hearing problem. We classify them into either prelingual or postlingual. Prelingual, they develop the condition before the age of language development, and postlingual, they developed after the age of language development. We group them into two. We group them also based on the number of people affected in the family. So if you have more than 2 people affected in the family, we put you into the familial cases. And isolated cases, we were able to get only one person is affected in the family. Then we took samples from the healthy controls too. We extracted DNA and used next generation sequencing and targeted sequencing to look for variants. So this is a picture showing us our sampling in the school. When we go to the school, we obtain administrative clearance, then invite parents to come. We sign informed consent. After that, we do audiological assessment for the participants, both the hearing and hearing impaired participants. After that, we take blood samples. We want to sample at least 2 unaffected people from each family. But when we invite them to the schools for the deaf, they usually would send an elder sibling or one parent will come. So we have to travel to different parts of the country to recruit the rest of the family members. Before the WACCBIP van came, we used to travel in this kind of vehicle, which looks like a death trap, to very remote parts of Ghana to sample families. So what did we see? From the students that were in the schools for the deaf, majority of them have prelingual hearing loss, meaning they developed the condition before the age of language development. And a lot of them had their first comprehensive diagnosis between the ages of 6 to 11. So this means that the parents knew that something was wrong with their kid, but they don't have the center, or they don't have the resources to test them until they are ready to go to school. It's mandatory and compulsory for them to get tested before they are admitted in the schools. And the schools some of the schools have the facility to test, so they get tested when they are going to school. Then when you look at the cause of hearing loss, a lot of the prelingual cases are congenital, which means they were born with a condition. But when you look at the postlingual, they have illness such as meningitis, cerebrospinal meningitis, causing the hearing loss, and they also have convulsion. With the convulsion, the parents don't know what caused the convulsion, but they knew that their child convulsed and became deaf. Then meningitis, we compared our data with what has been published in Africa, and meningitis remains to be a major cause of environmental hearing loss in Africa. But when you look closely to the story of Ghana, you would see that in 1988, you have high proportion due to meningitis and also due to measles. And this reduced in 2019 when we conducted this study. And this is good to highlight because it shows that our health system is working and we are able to give immunization to kids and prevent some of these diseases. So now let's look at GJB2. I told you I'll tell you a story on GJB2. So GJB2 is part of the family of proteins called connexins. And the founder variant that was identified within the Ghanaian population in high frequency is not exclusive to Ghana. So on this map, I'm showing other places that the variant has been reported. And I'll talk about the Japanese population in the next 2 slides. So Ghana, because we have the high prevalence of this variant, Ghana is the best place to study this variant. So we looked at our cases based on how we classify them. And the familial cases seems to have higher prevalence compared to the previous studies. This is because the previous studies conducted used random sampling, but ours was more purposive. So we had higher numbers. But for the first time, we reported a carrier frequency within the population to be estimated at 1.4%. This means that these people within the normal population have the variant, the founder variant. They are hearing, but if 2 carriers get married, then they have the possibility of giving birth to a child with the condition. To help solve this, we designed and developed a diagnostic tool, and this was based on restriction fragment length polymorphism. We used the restriction enzyme NCI1, which is able to recognize the DNA and cleave at specific points, and give us this kind of band pattern for genotyping. So we went back to the village with a high prevalence of deafness, sampled from there, and used our tool to test. We compared our results with Sanger sequencing, and we found out that our tool was very specific, and it's good, and it will be able to help us to genotype deaf people within the population. I want to say here that knowing that a quarter of the families we studied have this variant, it means this tool will be able to help us screen at least a quarter of the cases that we have, and it's going to be cheap because it's based on RFLP, which is not as expensive as sequencing. Knowing that the founder variant is present in other populations, we wanted to see whether we have the same variant, from Ghana traveling all the way to the other populations, or it occurred independently in the various populations. So from the Japanese study, they estimated the age of the variant to be about 6,500 years ago. So we also decided to estimate the age of our variant from the Ghanaian population. And our estimate was about 9,600 years ago. So it's older in the Ghanaian population than the Japanese population. So did this variant travel from Ghana to Japan? We use haplotype analysis to compare the haplotype across different populations to see whether we have the same haplotype. And the haplotype in Japan and other population is different from the haplotype from the West African region, and also the haplotype from the Ghanaian patients that we had. So it's clear that possibly this event was independent, and it occurred at separate times independently of each other. We also identified a variant that results in a stop codon in the GJB2 gene. And this variant has been reported in Mexico. So I did not need to do the protein modeling. The model was already published by these guys in Mexico. We screened other connexins. We looked at GJC3. We also looked at GJB6. We did not find any variant, but we found a pathogenic variant in GJB4. And for us to examine this variant and see whether it might or it may account for the hearing loss in this particular family, we did what we call the protein sequence alignment. And when you do the sequence alignment, you are looking for the conservation at that position that you are studying. So here we are looking at position 119, and you see that the amino acid at this position is conserved in all the organisms that we studied, which means that position is important for the protein structure and also for the function. So we modeled the structure, and we saw differences in the wild type and the mutant structures. Now, let's look at the next generation sequencing part, the interesting part. I want to say here that we have a lot of stories that we can tell here, but I will tell you only 2 of them because we won't leave here if I want to tell you everything about this part of the work. And forgive me, the slide is busy with a lot of diagrams, but I'll walk you through briefly. There's families that were negative for GJB2, we had 51 of them. We whole exome sequence them. So we're doing this in a tier kind of fashion, where we'll do targeted sequencing first, then next generation sequencing, exome sequencing, then the ones that we were not able to find a variant, then we do the genome sequencing. So with 51, we were able to solve 95% of them, combining both the targeted and the next generation sequencing, and this is very high. I think at the time that we published this, we were the first to have this kind of high rate of solving families with hearing loss. We found, 26 of our families segregated with non-syndromic hearing loss genes, 1 segregated with syndromic hearing loss. I'll tell you about this one. Then 13 of them had genes that were both non-syndromic and syndromic. We found 7 novel genes, and when we found these 7 novel genes, we looked for their expression in the inner ear. 6 of them were expressed, all of them were expressed in the inner ear. 6 of them had models, mouse models, and the phenotype was deaf. So being the first time reported within human, it's made it easier for us that we don't have to go the length of deleting this gene and doing the mouse study. So these are the genes plotted on the Ghana map, and it's very heterogeneous. Almost every family had its own unique variant, but there are 3 genes that were prevalent in the population, and this is myosin 15, cadherin 23, and GJB2. These 3 genes accounted for about 63% of the families that we studied. Then myosin 15 and cadherin 23, from our collaboration with our partners in Mali, Senegal, Morocco, and not Morocco, Mali, Senegal, and Cameroon, myosin 15 and cadherin 23 remain a common gene that is going through all these populations. So if we want to develop an African-specific panel, we'll need to include these 2 genes. And we also have a North African panel that is good for North Africa, but we need to get a panel that would be for Ghana or West Africa as well. And I highlighted over here a gene in yellow. This gene is otoferin. This is the gene that is now in the clinical trials. And from our last meeting, we have good results from the trial. So probably our families in Ghana will be able to benefit from this clinical trial when it is done. The common syndromic deafness that we found in Ghana was the Waardenburg syndrome. It presents in primal, except for thyroid stimulating hormone, which was high in the proband. The second story I want to tell you is on SLC12A2. This gene is expressed in the SLC12A2. It's expressed in the stria vascularis. And it's reported to be associated with this syndrome, Gilchrist syndrome. And the exon 21 is important for the function of this gene in the inner ear, such that when you skip exon 21, it is not expressed in the inner ear in mouse and also in mammals. So we mapped all the variants that were reported in SLC12A2, and the ones in black are the ones associated with hearing loss. And you find that a lot of them are within the exon 21, which means this exon 21 is very important. And the variant that we identified in the Ghanaian population is also in this exon 21. Here, this is a family that had the SLC12A2 variant. And when you look at the audiogram, all the affected people have from severe to profound hearing loss, and the unaffected people are normal. They have normal hearing. At the same time that we found this, our collaborators identified a family in Pakistan. So we collaborated with them, and this is the Pakistani family. All the affected people still had severe to profound hearing loss. So we went ahead to model the structure for this particular protein and found differences between the wild type and the mutant structures, especially when it comes to hydrogen bond formation, which is shown here on the slide. We did functional assay, expressed the gene in HEC293 cells. The expression data shows that the mutants had higher levels for SLC12A2 expression compared to the wild type. And we think that the overexpression of the mutant would stress the cell. So we went ahead and assayed for phosphorylated P38, which measures the stress level of the cell. And it shows that the mutant cells were stressed and they were dying due to the overexpression of the gene. Then with the cell morphology, the wild type had this axon-shaped cell morphology, which was absent in the mutant. So we think that our variant is affecting the structure of the protein and also the function of this protein. So to summarize everything, the major contributor to environmental cause of hearing loss in Ghana is cerebrospinal meningitis. And 3 main genes can be put into a panel to solve about 63% of families. And these are GJB2, myosin 15A, and cadherin 23. And I want to mention here that all the sequences that we had, we've submitted them so that we would help solve the problem of underrepresentation of the African genome in the genome database. We've also submitted all our variants to CleanVar and CleanGene to help improve the gene pair curation and also gene phenotype curation as well. We designed a tool that we can use to screen hearing loss within the Ghanaian population. Then we wrote a policy paper to help guide policy in Ghana. We have newborn hearing screening program in Ghana, but we don't have the genetics as part of it. So we are suggesting that they include genetics screening as part of this newborn hearing screening program in Ghana. And the tool that we developed will be very handy here. So I want to spend the last few minutes to talk to you about my work in the NIDCD at the NIH. So in NIDCD, I work with 2 groups, the Friedman Lab and the Hall Lab. And in these groups, I work on proteomics and immunoprofiling of patients with hearing instability. So these are patients who have fluctuations in their hearing, and we really don't know the mechanism. So I'm working on that to find a mechanism that results in fluctuation in their hearing. And this is a common phenomenon in the American population, but not in the African population. Then I'm also working on a mouse model, and this is built on a variant that was identified in the Pakistani population. So I want to understand the biology of or the mechanism by which this gene causes hearing loss. And also the overall goal is to develop a gene therapy for that particular gene. I want to thank my supervisors for the mentoring and thank all the head and staff of the hearing of the schools for the deaf in Ghana, and also all the participants, the parents, the students, who gave us their samples. They trusted us with their samples, and we are very much, we appreciate their effort. So the next phase of the project is to get the results we have, to feedback the results to the community. And the team in South Africa, working with the WACCBIP communication team and the WACCBIP team, is trying to find the best way of getting back the results to the various families that were sampled in Ghana. Thank you.
Thank you, Samuel. So I think we have time to take quite a bit of questions.
Morning, great talk. It's interesting that the children with hearing impairment, they have blue eyes, right? Is there any link with, high color genes and their hearing genes?
Okay, thank you so much for that question. So with the blue eye, the gene that is responsible for the coloration of the eye, the pigmentation, it also has something to do with hearing. So the same gene, when mutated, affects the eye and also skin. That's how come we see some of the pigments on the skin also losing, some of the cells in the skin losing their pigments, and also the premature gray hair. But to broaden your question, the eye is connected to the ear as well. So the vestibular part of the ear helps us to balance, and it works with the eye. So when you close your eye, you don't have 100% balance. And when your vestibular piece is also not functioning, you can also not balance very well, even when you have your eye open. So they work hand in hand.
Thanks, Samuel, for the great talk. Awesome talk. Good for I was wondering the plans for your clinical trial.
Can you say that again, please?
I was wondering the plans for your clinical trial.
The clinical trials.
Yes.
Plans for clinical trials. Yeah, I think we will get there, but we are not, we will do the animal studies first. So we do animal trial first. When we are done with that, then we can move into clinical trials. But clinical trials for hearing loss is quite expensive to see. So we will get there.
Okay. Thank you very much for your talk. I think you show clearly in your data that, meningitis is the main cause of loss of hearing in your study in the young people. So I'm wondering, did you try to also investigate in, let's say, adult people, mainly in old people? Because, sorry for my ignorance, it looks like when people are growing, when they are getting old, they lose also hearing. So did you try to investigate what is the cause of loss in hearing in this population? Thank you.
Yeah, thanks for that question too. Yes, so in the future, we have plans of doing age-related hearing loss. But currently, our focus is on the pediatric hearing loss, because if we are able to diagnose early and give the correct intervention, we will be able to help them with their cognitive development. And there is one thing I did not talk about it in this presentation because of time, but what we found in the Ghanaian population in Mali, Cameroon, is that these children with hearing loss are born to hearing parents, and the parents are not able to communicate with them. And we don't even have infrastructure. Our schools are not built for the hearing impaired. So it affects their quality of life. It affects their relationship with their families. And so on. So we are much more focused on the pediatric first, but in the future we'll be looking at the age-related hearing loss.
Okay, Dr. Dinko.
All right. Thank you very much, Samuel. Very nice talk. My question is with regards to the time of development, how you determine the time of development of hearing loss, whether it's postlingual or prelingual, because at a certain point, the people concerned cannot actually hear. Did you ask the parents or you did a test? The second question is, did you find any relationship between this time of development of hearing loss and the severity or the level of the hearing impairment? Yeah.
Okay, thank you. So with the prelingual and postlingual, so we look at people who develop the condition before age 2 into prelingual. And actually there is another phase between the prelingual and postlingual, which is the perilingual. That is between 3 to 4 years. But we combine perilingual and postlingual together to be postlingual. So anything from 2 years above, we put into postlingual. Yes, so the by age 2, the child should be able to make some sound to call mama, papa, and things like that. So some parents or all the parents know that my child was not able to make these sounds, will not respond to sound before this age. Oh, no. So that helps us to classify them. Then your second question, whether there is any link with severity. With what we noticed is that people with postlingual hearing loss tend to learn how to speak because it's postlingual, especially those who develop the condition after the age of 10 years old. There is one particular patient that I sampled, and she did not know she was deaf until she was in high school. And her uncle came home complaining that, I met you in town, I was calling you, and you think you are now of age, so you can be arrogant. Then she said she couldn't hear. So it was after then she began to take note that when people are talking behind her, she cannot hear. So she's able to read the lip, but not able to hear sound. So that's what happens with the postlingual.
Okay, thank you. I think we have a question online. It will be projected on the screen from Dr. Megache. So apart from the genetic mutations of connexin, especially GJB2 and its variants of the inner ear, are there some genes affecting outer ear defects? And can other factors apart from genetic contribute to hearing loss? Well, I think.
Yes, please. So there are other genes that are affecting the vestibular part. So people who are studying balancing and spatial orientation of the head are able to come up with genes that affect the vestibular part of the ear. Both the vestibular part and the cochlea, they have hair cells, but the hair cells are connected to different neurons to the brain, and they perform different functions. So we have genes that are expressed in the vestibular piece. And yes, we have different causes to hearing loss. We have the noise-induced hearing loss. We have ototoxicity, which is due to use of drugs like anti-cancer drugs and antibiotics. We have sickness or accidents also causing hearing loss.
Okay. Okay, so I think we'll take one quick question from Chinedu and one more from Nikes, and then we'll move on.
Okay. Thank you, Samuel, for the presentation and for the work you're doing. I just have two questions. One is on the mechanism of hearing loss and pathogenesis. Is it the same, regardless of whether genetic, environmental, is the pathogenesis the same? Then the second question is on the gene, the GJB2. Is the gene only functional or associated with hearing loss? Does it perform any other function in other parts of the body? And the protein, so when you look at the protein, what happens? So is the protein not expressed, or the defect is only at the genetic level? Thank you.
Okay. So the mechanism for environmental hearing loss and genetic they are different. They all affect the ear. Even with the genetics, different genes have different mechanisms to which they affect hearing loss. So, for example, someone who has noise-induced hearing loss might have some of the hair cells dying at certain frequencies. And this is also common to orthotoxic hearing loss. But someone who suffers an accident can have the bones in the inner ear displaced, and that can also cause hearing loss. With the conductive, some children are born with a flesh covering the whole ear. So they have to do surgical procedure to remove the flesh for them to hear. So the mechanisms are different. Then with GJB2, yes, so the expression, I would say that depending on the variant, some variants result in reduced expression of the gene. Others will not express the gene at all, especially the truncated ones. You won't have the protein to perform the function. Some of the variants, you have the protein all right, but the protein is not functional because it's supposed to form gap junction to allow for cell-to-cell communication. But if this junction is not formed, then the cells cannot communicate.
Yeah, very nice talk. Yeah, my question was just to expand the previous questions. So there is a growing interest now in the North to explore epigenetics in hearing loss and many non-communicable diseases. Do you have plans to explore, to extend and investigate that in Africa?
Yes. Yes, so the study is still continuing. We are still looking at patients, and we plan to extend the study. So the collaboration between, this work is a collaboration between WACCBIP and Hygienes Africa, and the collaboration is still going on, so we'll still be doing more.
I think we can all agree that was a great start to day 2. And to continue on from there, I'm going to invite the chairs for the next session, which will be molecular epidemiology and enhanced epidemic preparedness. And these will be our very own Dr. Elma Amatepe and Dr. Lucas Amenga Etigo, and they will take us through to the next session.
Hello everyone, good morning. It's good to see you all this morning. So far we've been making some very good time. So at this point, we'll hope and ask our next speakers to emulate our keynote speaker and try and keep to time. First on the list we have Chinedu A. Ugu, and he's going to talk on the pandemic preparedness post-COVID-19 in Nigeria, African Rapid Immunosurveillance System for Epidemic Response Arise. So Chinedu, the floor is yours.
Good morning, everyone. I just hope I can keep up to time. That's the problem.
Try.
So I'm going to tell the story of African Rapid Immunosurveillance System for Epidemic Response called ARISE. My name is Chinedu. I'm a senior lecturer at Redeemers University and a postdoctoral fellow at the African Center of Excellence for Genomics of Infectious Diseases. And I'm currently at the NIH as an APTI fellow. So I'm going to start with telling the story of SGID. I guess most people, if you don't know, so SGID is the Center of Excellence, more like the sister of WACCBIP in Nigeria. So SGID was also founded in 2014, so we'll be also doing our own anniversary this year. So SGID, we were very instrumental during the Ebola outbreak in 2013-2014. And then for SARS-CoV-2, we provided the first genomic sequence of SARS-CoV-2 from the continent. And so far, we've sequenced over 10,000 SARS-CoV-2 genome across 26 countries in Africa. And currently, SGID is a reference lab for genomic sequencing for COVID-19. So the story of ARISE, this is the summary. So in 2018, I was finishing my PhD. I moved back to Nigeria with support from the BBSRC on a project called One Health Accelerating Vaccine for Ebola and Lassa fever. This was a collaboration between SGID and Nigeria, and the aim of the project was to study immune responses to Lassa fever in Nigeria. So while we were doing that work, then COVID happened in 2019. And so most of the capacity we built with Lassa fever, we translated into working with COVID, and which we got a good seed funding from Cambridge, Africa, on the project SARS-CoV-2 Immune Correlate of Protection in Nigeria. Then with that effort, we also got some good support from the Gates Foundation to then set up the main project, which is what I'm going to talk about, the Africa Rapid Immunosurveillance System for Epidemic Response. So the main objective of the ARISE platform is mainly, one is with the enormous progress we've made in genomic sequences. So it's more like providing immunological data to support the genomic sequences. And then we hope that most of those immunological data we'll share with public health agencies like the NCDC in Nigeria, the Africa CDC. And then secondly, in one of the things we found that was a struggle during COVID-19 is having curated samples. So another aim of the ARISE platform is to curate sera PBMC from mostly emerging pathogens that you have endemic in the continent, and then keep them, should in case there will be any pandemic or epidemic, we'll provide this sera for research and for interventions. So also, we are hoping to be to be a center of excellence for immunological research and then training. And then all of this data we generate, we ensure that we share them in real time with all the public health agencies. And then to mention here, the whole essence of the ARISE platform was built on collaboration, both within the continent and then outside the continent. So this is what the platform looked like. So we have antibody assay where we use both ELISA and then Luminex. And then we do, to measure binding antibody response, we use viral pseudotype neutralization to measure neutralizing antibody response. We use ELISpot to measure T-cell functional assay, and then we use flow cytometry to do phenotyping. And then we also do single cell and B-cell and monoclonal antibody discovery using RADpig. So I'm going to start with two examples of the work we've done with ARISE. So starting with Lassa fever. I don't know if people don't know Lassa fever. Lassa fever is one of the viral hemorrhagic fever that is endemic in West Africa. So it's one of the old world arena viruses. It's genetically diverse. There's no licensed vaccine for Lassa fever, but there are two vaccines that are currently in clinical trial. What the gap in Lassa fever is that it's one of the neglected tropical diseases. So the immune correlate or the features of protection is not yet defined for Lassa fever. So one of the work we did is with the OVO project is to define some of this, the type of immune response you see from patients with Lassa fever. So the first thing with some of the data, one is here, we measured T-cell response from Lassa fever survivors in Nigeria, and then the contact. And these exposed contact are the people who manage Lassa patients. So you could get Lassa from contacts and also from the rodents. So we recruited survivors of Lassa fever and their contact. And these contacts include those who take care of them, the family members, the clinician, and the nurses. And the interesting thing we see measuring their T-cell response, and here we use the nucleoprotein of the virus and then the GP as the antigen. What we see is that both the survivors and the contact who never tested positive for Lassa fever, we are making the same level of T-cell response. And also we could show that the major T-cell response is coming from the NP1 region of the nucleoprotein and then the GP2 region of the nucleoprotein. We also measured the binding antibody response using ELISA. And again, what we saw is that those who didn't even test positive for Lassa, which are the exposed contact, and then those who tested positive, again, had similar level of response. So, but where it becomes interesting now is when we then did the neutralization assay. So we have pseudotype viruses that expresses the different GPC of the different lineages of the Lassa virus. So we have lineage 1, 2, and 3, and 4 of the Lassa virus. And then what you then see is that it's only the survivors who made neutralizing antibody response, while the exposed contact do not. And this is simply because the survivors have symptomatic disease. They are clinical cases, while the exposed contact, they are asymptomatic. So we think they don't have enough time to make neutralizing antibody, even though they already have the binding antibody response. So then, when COVID happened and then we got funded, we basically translated everything we've done for Lassa fever COVID to understand the immunological responses from different COVID cases. So for COVID, we recruited the different doses of the vaccine cohorts. Sorry. And then we also have the people that survived COVID as well. We also have the exposed contact. One special population we have is the pre-pandemic samples. So these are the samples we collected from Lassa fever. And then when COVID happened, we also tested them. And then we have the breakthrough, and then we have the non-negative control. And these are the people we've taken time to create and ensure that they are negative for most of the endemic pathogens we have in the continent. So for COVID, the take-home from our COVID assay result is we also did binding assay and then neutralizing assay. And what you see with COVID is that both the exposed contact and then the survivor, they make the similar binding antibody response, and they make the same level of neutralizing response. And this is, I think here we tested the different strain, the Y type, the beta strain, the Delta, and then the Omicron BA1 at the time we did this work. So different from Lassa, COVID people, whether you were asymptomatic or symptomatic, you do make both binding and neutralizing antibody response. And then this is one interesting thing we found, and which we hopefully other people also have reported it, is that the pre-pandemic serum, these were the people that were samples we collected pre-COVID. And you see that they also have the same level of binding antibody response with both the convalescent COVID-19 patient and then the contacts. But the interesting thing about this is that this pre-pandemic serum, they didn't have neutralizing antibody response. They only had binding. So this probably will be from the old coronaviruses that is different from SARS-CoV-2. So now, this is the summary of everything I will tell you. So the key thing we'll do, like I said, we generate all of this immunological data and we'll share them in real time. But one thing we try to do is, again, to curate some of these samples, both the PBMC, the sera, should in case there's any outbreak. I think so far we have about 540 samples from Lassa survivors and then about 650 from SARS-CoV-2 survivors. The other part I'm doing now is I'm currently at the NIH, and my project at the NIH is mainly isolation of monoclonal antibodies from these Lassa survivors. So most of these samples that we've created, so the aim now is then to isolate monoclonal antibodies from them that you can then use for diagnostics and therapeutics. And again, about the training, so far from 2018 to now, we've graduated about 8 MSc students and then 12 undergraduates. So those are some of the outputs from the work we do with the RIs. Then the current work ongoing, now we've expanded the RIS program, and we now have also other funders beyond the GATE and then the Cambridge Africa. So we have different projects ongoing. Like I said, I'm at the NIH doing the isolation of the monoclonal antibodies. These are some of the PhD students we have at the at SGIT and some of the master students working specifically on the RIS projects. So we have projects on other projects on Lassa fever, which we are trying to understand the bottleneck between transmission. So bulk of the Lassa fever infection comes from rodent to human, but some cases in the hospital where you have hospital transmission, nosocomial infection. So what we want to understand is why those pathogens manage to do human to human versus the normal route of rodent to human. So we have a PhD on that. We have another PhD student who is now looking at the correlate of protection from Lassa fever. We also have other viruses that we study. We're looking at epidemiology of HIV, HPV infection. So these are some of the ongoing work on the ARISE platform. So I won't say all of this without telling you the challenges. So I could say, oh yeah, everything worked out perfectly for us, and then we are doing very well. But no, we do have we also have our own successes. So one is funding support. You also struggle to get good funding. And then one of the challenges, and we tried our best in correcting it, is where when you do collaboration, where people are more interested in you sending samples to them without building capacity. And that is one thing I think we've made progress with, ensuring that whatever collaboration we do is either proper beneficial to both team, both the collaborator and us. And the other challenge we talked about, and I think everybody will have this problem, is lab supply. By the time you get samples from the whole, by the time you get samples to work, it may take 6 months to get the reagent to do the work. And then I don't know if you guys also have this problem, but retention of talent. In Nigeria, we call it the Japa syndrome, where every young, talented PhD student wants to travel abroad. And I don't think I should stop them from going abroad, but I do think we should find a way to, whatever is attracting these young people, talent to abroad, we should find a way to retain them here, and so that they will also continue to legacy of doing excellent research. So I think this should be my last slide on this. I'm going to thank all the funders and all the supporters we've had over time. First of all, my supervisor in Cambridge, who has enormously supported me and ensured that I set up, and also my director at SG, Christian Happy, and the funders, Cambridge Africa, who believed in us when we think we're not doing anything. And then the Bill and Melinda Gates Foundation. I also will thank the NIH that is helping to then take us to the next stage, which is translational immunological work. And every other person that we've listed that I don't mention. So lastly, I will say thank you, and I could take questions. I just hope I'm keeping to time. But I will also use this opportunity to invite you guys to our own anniversary, which will be in October, and we'll be launching our new genomics center. So if you've enjoyed Ghana Jollof, don't worry. You know you have a good time coming to Nigeria in October, and I think at least now we'll take questions. Thank you.
I was going to commend your very good timing, but you had to bring in the Ghana Jollof. But that was very good timing and good presentation. Thank you so much. We'll take questions. Peter has his hand up. Yes, so Dr. Kwashi.
Hi, Chinedu. Great talk. And I'm happy that you finally agree with me that those cross-reactive antibodies are not neutralizing.
I've always agreed with you.
Okay, so I had a question about your Lassa samples where they are asymptomatic. Do you think it's because there wasn't enough viral load in the initial infection to actually have a productive infection, so the immune system is exposed but not really, the virus is cleared before it actually starts anything? Is that what you think is happening?
Everything you say is correct. We don't know what is the actual mechanism, but that is one is probably they didn't have enough viral load. Or it could also be true they are genetically diverse, that they're able to mount a good response. So I don't think I mentioned it, but I could say that in Lassa fever, 80% of the infection are asymptomatic. So the problem is that the 20% that are symptomatic, the fatality is very high. So that's why it becomes a big problem. So when you look at this, a lot of people will carry antibodies. So you say that, one, you're correct, that the infection is not enough to have mounted a good immune response. So those people will survive, or they have good immunity that they were able to clean up before it becomes symptomatic, or it's genetic. So the answer is we don't know.
Hello, Chinedu.
Yeah.
Thanks for the presentation. So, how did the immune response play out in COVID, knowing that Lassa is already existing and there could be comorbidity with COVID? Did you look at that?
That's one of the things we want to look at. So you see what that was the idea we want to test those Lassa samples. So we first of all thought, okay, this is endemic, and then COVID happened. Do we see anything? And then which is when we saw the binding antibody response. Oh, okay, people already have this. So we then do neutralization, we didn't see anything. So part of the thing we intend to do is to go back to our Lassa cohort, because that's one good advantage we have with SGIT, is that we have a clinical site supporting us. So to go back to our Lassa cohort and see those of them that had COVID alongside Lassa and how they fared. So those are things we want to do in pipeline. Thank you.
So thanks, Gina. I enjoyed your presentation very, very much. And I think you have a treasure trove, you know, and you mentioned at the tail end that the focus of your postdoctoral work is actually to try and, you know, isolate monoclonals. And my question is, what are your plans now to exploit this treasure trove and make diagnostic products and therapeutic products? It's very exciting to me to see that that's part of the plan.
Well, you are my plan, you know. Oh, okay, thank you. I think you know you are my plan. So it's a good, so for us, again, the end game or the major objective for everything we do with immunology is to get to the level of translational. It's not just how do we get this to the people? So part of the work I'm doing at the NIH is to isolate the monoclonal antibody, and then hopefully once we get them is to do clinical trial and see how we can get them into the market. There is no, like I said, there's no licensed vaccine for Lassa. And one of the set of people that are very endangered or vulnerable to Lassa fever are the healthcare workers, the clinicians. They are usually the most affected. So if we can manage to get a therapeutic antibody that they could take, that they can, when they handle some of those patients, I think that will be a game changer. And I think this is what I'm hoping to. Once we get this monoclonal antibody at NIH, which we are making progress, it's just that the NIH is not part of the presentation. So once we get this antibody, is how to then get them to the people. So you are part of the plan, you know.
Yeah. All right. Yeah, thank you. I have two questions. Yeah. One question is, I want to know which variant or which lineages you use for the neutralization assay for the Lassa. One. The second question is, can you elaborate on your collaboration with Enfield Liberia, please? Thank you.
So I didn't get the second question.
Your collaboration with Enfield. You mentioned Enfield as your collaborator in Liberia. So can you elaborate on that, please?
In Liberia?
Yes, you mentioned.
Oh, okay, thank
yes.
you. Yeah, sorry. So it's early morning. I don't work well in the morning. So the neutralization. So we have. This is the interesting part that this is the capacity we are doing very. For this, the work I presented, we use the GPC of lineage 1, 2, 3, and 4. So that's what we presented. But I could tell you now that we do have capacity for 17 lineages for Lassa. So when I say 17, it doesn't mean that you have 17 lineages circulating. So some of them are not real lineages, but we hope that we should be able to cover both the existing lineages and those that will envisage that will happen in the future. So we have capacity to test all of them. But for the work I presented, we've only tested 4 lineages. So the collaboration we have in Liberia is with, I think you saw it at the acknowledgement. So that's with EXIT. So that's the genomics work that we do. So we do support most African countries, Liberia, Sierra Leone, Guinea. So I think I won't be the best person to comment on that. Christian Happy will be the best person to comment on that. But those are the genomics work that we do. Thank you.
Yeah. Yes, yeah. There are more questions too.
Jesus.
Yeah. Yes. Yeah.
Right?
Thank you.
Yeah. Yes.
So
I'm here.
Okay, there you are.
Thank you so much for the wonderful presentation. It's interesting to see the Lassa fever patients and their close contacts. You have similar antibody levels. Do you think the differences in neutralization ability is mainly ability? Yes. Yeah. So what I'm asking is, between your patients, the Lassa fever patients and their close contacts, you have similar antibody levels. I'm wondering if you've investigated antibody ability subclasses, maybe if that could explain why you have different neutralizing abilities.
I didn't get it properly. Or the subclasses of the different antibodies?
Yes.
So no, we haven't gone to that level yet. We haven't classified it. So what you are seeing here, the binding antibody is IgG. So it's only IgG that we've worked on. So we haven't looked at the different subtypes of the IgG yet. No.
Okay. All right.
But I do think, like, which is what Peter alluded, is why we do think is that the contact, even though they are not testing positive, they must have had the virus at some point. So they may have had asymptomatic cases. Their body do have this whole signature of immune response. Now, whether that immunity is what is protecting them or it's just there, we don't know yet.
Definitely we'll talk further after.
Thank you.
Thank you very much.
Next presenter will be our own Dr. Yawaniwe. See here.
It's online. Yeah. Can someone, Martin, check in the office? Maybe he's forgotten. It's communications here. If you can play some of our sponsors stuff that whilst we wait. 2 minutes. If you don't find him, we'll change. Video. Yeah. I don't even know. She's saying that. And then we are going to the fellow sessions. Oh. Communications, you guys.
Akwaba to all my lovely, lovely friends from Ghana and in and around. So thank you so much. I'm actually not from Ghana, as you can see, right? Well, we are all a big bunch. We're all a big bunch of scientific geeks that have come here to explore and learn and evolve. My name is Vikas, and I'm from Mumbai, India. And from India, I greet you all with Namaste. That's what we do, right? So thank you so much. I've traveled all the way from a city called Bombay. I'm sure Mumbai is a city that you guys probably heard of. It's about 8,000 kilometers away. And this has been a very exciting 2 days for me. I got in 2 days back. And let me tell you, I don't feel I'm 8,000 kilometers away. I feel I'm very much in my home, in like India, because the Ghanaian hospitality, the people of Ghana, really, you guys have touched my heart, touched the heart of every Indian. Thank you so much. Right. And from the time I landed into this beautiful country at the Accra airport, I was greeted with a smiling officer at the customs, and she smiled, and I don't know why she was smiling, and everyone and everyone that I met has been smiling at me in Accra. So maybe I'm like the Tom Cruise out here, I don't know. But lovely people, guys. You guys are lovely people. Let's give a round of applause to all you people. So I'm actually dressed in the WACCBIP dress as well. I'm going to take this back and I'm going to show this to all the scientific community back in India. We have a big scientific community in India. And I'm going to show this to them. This is what Ghanaians do. This is how they make an event successful, right? So thank you. I'm not going to take much time. Of course, this is a very different presentation. It's about our company. It's about how we add value to science and research. And I'm just going to take a little time of yours, maybe 5-10 minutes. And if I run out of time, you can just tell me to stop and I will get out of here, okay? But it's a scientific conference. So apart from learning and absorbing different scientific research papers, it's very important for all of us to understand the value of the vendors, the people who provide reagents, the people who provide all the stuff for your laboratory work. And we're one such company doing this for the last 50 years, yeah? This is a family business. It's actually 47, I'm sorry for that, but this is a 50-year-old company. We're called SRL, Cisco Research Laboratories. We were the first company in India that started manufacturing molecular biology and bioscience products. The story goes like my grandfather was a very poor man back then, but he had a vision and he was invited by the President of India to start manufacturing products because the President of India told my grandfather, why should we import from Sigma, Merck and Thermo? You know, that's the Indian story, right? We have a lot of respect for these European and American brands. But the president said, you know what, I will give you $500. He told my grandfather, I will give you $500. Why don't you set up a laboratory? And that's what we did, because we didn't have, my grandfather was a businessman. He didn't have money, but he had the vision to make a big company. And with $500 of money, today we are about $45 to $50 million revenue company selling products in more than 100 countries. So we started manufacturing with amino acids and, yeah. And let me tell you, we sell to more than 40 countries within the African Union as well. So we are a very big presence here. But anyways, I'm going to move ahead now. So our motto is the endless process of science. And what we do is we manufacture molecular biology, American Chemical Society grade, that's called the ACS grade reagents. We're obviously very strong in pharmaceutical and biotech products. As you know, India is a hub for manufacturing the formulations, the drugs, the vaccines. And our company provides raw materials and biopharmaceutical products to companies like Sanofi, to companies like Pfizer. They have huge operations in India. Of course, in Europe we sell as well, but we have a very big base of pharmaceutical products. There's only 1 or 2 companies in India that manufacture culture media for microbiology. So we manufacture our own dehydrated media. We do raw materials. We have animal houses. We have our own peptones. And we are the first company that manufactures DNA and protein tools. So all the COVID engulfed our lives in 2020, right? And as you guys have fought the virus here in India, we were doing the same. And our company was the first company to start manufacturing all the reagents which were used by the diagnostic companies that started selling off the kits. So we've played a very pivotal role in Southeast Asia to provide diagnostic reagents to all the kit companies, right? From Korea to China to India to even the Gulf states. So we have a company that's another division called BioLit, where we make a lot of Taq polymerases, our reagents for DNA and RNA extraction. Well, not going to explain much, but they're used for quality control, analytical development, and as I mentioned, pharmaceutical and biotech manufacturing, right? So this is a little bit about us. We have 3 manufacturing locations. In India, we have 5 distribution warehouses. We are a reputed company. We have the ISO 17025 certification, which is a quality certification. We are one of the few Indian laboratory chemical companies that have FDA and CGMP. There's not many Indian companies that do the FDA registration for lab chemicals. We have, of course, maybe more than 200,000, 300,000 scientific papers. We employ about 300 people on payroll, but about 200 people on contract. So in a nutshell, we're a company giving jobs to more than 500 people. And let me tell you another interesting thing here. For all the lovely ladies here, in our organization, all the top positions are held by women. You know, we have, yes, there's one manufacturing unit which is only controlled by women. Right from the sweeper to the head, there's only women. We do not allow even men to join the biotech unit, okay? That's our contribution to helping giving equal opportunities to all the ladies of all, the scientists in our country. And that's what we do. Of course, as I mentioned, we are present in the complete world. We have about 800 global dealers that are serviced by 120 distributors globally. We have about 8,000 products and about 20,000 stock keeping units. In short, guys, we're called as the mini Sigma Aldrich of India, because this is what the tag was given to us way back in 1975 when we were established. People called us the mini Sigma of India because we were the only company that had the quality that could match up, that could stand up to an American or a European reagent. And even today we are called as mini Sigma of India. That's why I'm here to present our products, to showcase our offerings out here. Of course, we sell for different purposes: semiconductor, biology, proteomics. You guys are doing a lot of research here in different departments at the UG and all the different places that the other students have come from. We are very strong in molecular biology, even for organics like HPLC, gas chromatography, solvents. We are the only company that manufactures nano powders in India. There's a lot of emphasis now on the increasing nanotechnology sphere. There's a lot of money being pumped in by different governments all over Europe, all over Africa, all over the Asian continent for innovative products, innovative drugs using nanotechnology. So we manufacture graphenes, we manufacture nanotubes, we manufacture lots of nano powders, which you could buy now from us since we are here. That's the idea. So of course, I'm not going to go too deep, but just a little run through of what we manufacture. We have a big range of HPLC solvents, which, any HPLC lab, they need the basic things like acetonitrile, methanol, N-hexane, water. We do a lot of these products. We do dried solvents. We do ion pairing reagents. We are very strong in organics. Of course, this conference is more to do with cell biology, but I'm just going to run through all the different products that we do. We have a big range of plant growth regulators, even precious metals, boronic acids. We are very strong in silica gels. I know you guys use a lot of silica gels even in the UG different departments here. We manufacture those in a very big scale. Let me tell you, we are the only company that have solvents which are molecular biology grade solvents. That means our products are DNase, RNase, protease free. I'm talking of solvents which are used for extraction purposes. Usually you would not find molecular biology grade solvents. So we manufacture the MB grade solvents which are DNase, RNase, protease free. They're very interesting. Very few companies in the world make molecular biology grade solvents, and we're one of them. Of course, biochemicals is a very big foray of products. We do various enzymes, substrates, detergents, sugars, amino acids. We are the pioneering company for molecular biology from India, and that's why the WACCBIP really interests me, because this center is all about biotech, diagnostics, molecular biology, cell biology. We have a huge range to meet all your different lab requirements. Electrophoresis. We are the electrophoresis leaders in Asia. I'm sure you do a lot of electrophoresis in your labs. We are the largest manufacturers of agaroses in Asia. We sell agaroses to companies like Sigma, to companies like Thermo, to companies like Merck. We actually sell our products to a lot of these companies. There's a slide that I'm going to show you guys after this. Of course, catalysts, DNA, RNA accessories, we have it all. As I mentioned, we are the pioneers of nanotechnology in India. We sell products for drug delivery systems, for research in different centers of research that are engaged in nanotechnology. Our medias are products that are used primarily for microbiology. As you know, you guys might be using products from companies like Oxoid, like BD, Diffco. We buy a lot of the raw material from the same sources in Europe where they buy it from, so our product quality is equally good, with, of course, a much, much, much more affordable price for our Ghanaian brothers and sisters. So, and I'm very sure about this, guys. We also supply to some of the labs here, and we would hope to have more business because we want to add more value to your work that you do. That's the real range of our protein tools. So we do reference standards, we do protein markers, we do columns. We have beautiful products. I mean, I'm going to actually have you guys distribute a little brochure so you guys can go to our webpage. Our webpage is like the Aldrich webpage. It's very advanced. There's a lot of information for all you students to learn and understand. So, of course, we are very strong in pharma ingredients. Of course, this is an institution, but institutions are the birthplace of industry, right? So there's lots of products that we do in bulk scale, And these are some of the specialty products. We do it in lab scale as well, but we do it in bulk scale as well. Vaccines has been a very, very big contribution from our company. All the big vaccine projects. I don't know if you guys have heard of an Indian company called the Serum Institute of India. The Serum Institute is the world's largest vaccine manufacturing company. It's bigger than it's bigger than Janssen, it's bigger than many of the companies in Europe. We are in a very strong partnership with the Serum Institute to manufacture, I'm going to tell you guys a very interesting story here. The COVID vaccine, okay? I think the COVID vaccine which we got, the Indian guys got, was made by the Serum Institute. It was developed by AstraZeneca in England. And it was a chimpanzee. I'm not a bioscience student, guys. I'm a business graduate, but I think last 15 years I've learned a lot. So excuse me if I'm wrong. You will not believe, Serum Institute is actually developing the world's first vaccine for malaria. I think that vaccine is still in it's in the trial stage right now, but or maybe it has come out. I'm not sure about what's the name? I'll share it with you. I will take my royalty for that from you. So the malaria vaccine was used. So what happened is November, December 2019 is we started hearing cases of COVID, right, from China and from other countries. Serum Institute already had a whole platform ready for the malaria vaccine, and they used the malaria vaccine platform to actually immediately divert their interest to the coronavirus vaccine platform. So if there was no malaria, there would be no COVID cure, for the Indian population, because we are 130 million people, 1.3 billion people in India. So it's very important that this company, the Serum Institute, is a very big consumer of our products, and we are very proud to be associated with all the different government platforms that help for COVID. In fact, the Fortune magazine in the US, they actually covered me as a COVID warrior in 2020 because we did some really good work. We tried to save lives indirectly, by developing kits for the coronavirus. So we're really proud that we got this opportunity. Well, a little bit about our labs. These are just our R&D labs. And I'm not going to go too deep in here, but this is some of the products, the way they're stored. We have more than 500,000 to 1 million bottles here that we stock. This is all our warehouses in India. We have 9 locations in India. We sell to more than 120 countries globally. And this is how our product looks. So our labels are pretty much like how you could have all the necessary information. Our entire family of products is here. This is how typically our label, they look like, and we have all the information like specifications, impurity profile. The difference between a good laboratory reagent and a regular normal laboratory reagent is how many companies give you the impurity profile. Everyone will say it's a 99% or it's a 99.9. But the main thing is the impurities. And we focus on the impurities because we're a company that consider Fluca as our Bible. Fluca Rydaldehyde, I'm talking of 100 years back when, I don't know where I was, but my grandfather, he was the one who took up the Fluca book, and he devised every single specification of SRL to be in accordance with Fluca specs. That's our power. We are the gold standard in the lab reagent business. Well, as I mentioned, we have huge number of distributors. We have a presence in the African subcontinent. You could see the countries that we have representation in. In fact, I'm so much in love with Africa. This is my 5th trip in like 1 year. And I'm again actually coming to Cape Town after 2 months for another show called MedLab. It's a diagnostic show. But I've started to love African people. I've started to love the way you guys are trying to build your country. Very much a story like India. We are exactly like you guys. We really worked hard. But I can see that Africa is the future of the world. It's the future of the world. That's a fact of life. It is. And I think I would like to tell you that we have a lot of African students in India, by the way. There's a lot of African students researching in different research centers in India. So there's a lot of friendship, of scientific knowledge share between India and African nations. So it's good. So these are some of our clients, guys. So like Dow, Abbott, BASF, Bayer. You can see the kind of companies that use SRL products. There's also global testing services like DNV, SGS. There's food companies like Coca-Cola, Diego, Heineken, Kellogg's, Kraft, McDonald's. We eat this stuff every day. We sell to all these companies. We actually sell to all the labs of all these companies. It's taken a lot of effort, it's taken a lot of convincing, but it has happened. Because, you know, we all want a Mercedes and a BMW, right? But actually the stuff what is inside like another car which is half the price is probably the same, but the BMW has the brand, right? So we're trying to build the brand, and we want to tell you that we are the brand for all your work. I've done, almost finished, some clients. These are some of the companies, guys, that we manufacture for. So you can see, I'm sure you guys buy your products from a lot of these companies for your lab work. And you won't believe these guys buy from us. It's a fact. I mean, there's nothing to lie about. So the many products they buy from us because they know us. We've been around for 50 years, as I mentioned, half a century. And our products, our quality is good. They take our products, label it into their brand, and you know that's how business works. This is how business works. The reason why I'm here, if you can buy and trust them, and if we are one of their vendors, why don't you can buy it from us as manufacturers, right? That's the whole idea. Because Africa, the same thing is you have limited resources of funding and you have good knowledge. So you got to save money somewhere, right? So we're there to help you save money. That's our objective, right? That's it. Thank you so much. Thank you. I'm really happy that I'm in your beautiful country. And 3 cheers for all Ghanaians. Hip hip, hooray. Hip hip, hooray. Hip hip, hooray. Thank you, guys. Thank you.
Sorry. Champion Makata. Thank you very much. I guess you all agree with me that this passes for a keynote lecture. This passes for a keynote.
I just want to add a global So he's our local distributor in Accra, Global Excelsior. We have a small booth back there. Please come and have a look. We don't have free coffee for you this time, but the next time we'll arrange one. but definitely, these guys are doing a great job.
Andy, we have 2 minutes. Thank you very much. So, as we mentioned yesterday, we have the exhibition stands there, both for the scientific and laboratory companies and also for the Ghanaian artifacts. Please make some time to visit them. They are just behind there. They have beautiful Ghanaian dresses and a lot of goodies you can take with you when you return. So we are going to have a very quick coffee break this right after my announcement, and then we'll be back in 15 minutes. Thank you.
10:40. Thank you.
Come back at 10:40.
sent them. Yeah. Oh, the woman who spoke, I did it yesterday, and I thought I had sent it. I just realized, so. Yes. So it's without it. Yeah. So there are two different files. Yes. The moment you sent the reminder WhatsApp, because I did it yesterday and I thought I had sent it. So the moment you sent it, I was like, oh, what? So immediately I just sent it. The person online come. You know it's online. You want to see it here? No, you can't see it here. And so you will check it because it's streaming, so we have to. You see, they don't use coupons for hello. Morning. No, I meant that. number them, which one is first, second, third.
That's the reason to we are not predicting. That's how that just have to walk you through.
Yes, the reason I'm saying that is usually in the layout of the program. You see the way by all of us, just the way this comes up with this. That's all I'm doing. This is first, second, third. Hi, Mark. Okay. Which one do you want? For Illuminus or for me? Okay. Okay, should I send it to you on WhatsApp? Okay. All right.
Get rid of the hassle of procuring lab supplies and start running and continue. Hello there, we're Excelsior Global Limited, and we are back with exciting procurement and supply solutions for you. Take a moment to imagine a laboratory where procurement was such an easy task. Our lab equipment and consumables are only of the highest quality, and Excelsior procures these from the most highly regarded manufacturers. Our lab supplies include equipment, reagents, and consumables that you use every day in your lab. Get rid of the hassle of procuring lab supplies and start running your laboratory smoothly. Take your lab experience to the next level with our game-changing solutions. Choose Excelsior. Hello there, we're Excelsior Global Limited, and we are back with exciting procurement and supply solutions for you. Take a moment to imagine a laboratory where procurement was such an easy task. Our lab equipment and consumables are only of the highest quality, and Excelsior procures these from the most highly regarded manufacturers. Our lab supplies include equipment, reagents, and consumables that you use every day in your lab. Get rid of the hassle of procuring lab supplies and start running your laboratory smoothly. Take your lab experience to the next level with our game-changing solutions. Choose Excelsior. Hello there, we're Excelsior Global Limited, and we are back with exciting procurement and supply solutions for you. Take a moment to imagine a laboratory where procurement was such an easy task. Our lab equipment and consumables are only of the highest quality, and Excelsior procures these from the most highly regarded manufacturers. Our lab supplies include equipment, reagents, and consumables that you use every day in your lab. Get rid of the hassle of procuring lab supplies and start running your laboratory smoothly. Take your lab experience to the next level with our game-changing solutions. Choose Excelsior. Hello there, we're Excelsior Global Limited, and we are back with exciting
should I give it? What's your name? No, I mean, it doesn't matter. It's not enough proof of security access. She just so if she wanted a key, but if it was a guy, he would have failed. Because it's a lady. But she came for it earlier. I don't know. She came for the key earlier. I think so.
Hello there, we're Excelsior Global Limited, and we're back with exciting procurement and supply solutions for you. Take a moment to imagine a laboratory where procurement was such an easy task. Our lab equipment and consumables are only of the highest quality, and Excelsior procures these from the most highly regarded manufacturers. Our lab supplies include equipment, reagents, and consumables that you use every day in your lab. Get rid of the hassle of procuring lab supplies and start running your laboratory smoothly. Take your lab experience to the next level with our game-changing solutions. Choose Excelsior.
WACCBIP came out of the energy of many young people in the department at the time.
WACCBIP's excellence really stems from teamwork.
We are at the cutting edge of technology.
We make excellence the bar for training at WACCBIP.
We believe in excellence in teaching and excellence in research.
To do high quality science, to be able to publish and to make an influence in the scientific world.
The West African Center for Cell Biology of Infectious Pathogens, WACCBIP, began in 2014 as one of the World Bank African Centers of Excellence, located in the University of Ghana, Legon. In 10 years, it has grown into a leading center for the training of the next generation of African scientists and is spearheading groundbreaking research into infectious diseases, genetics research, and research into non-communicable diseases like cancer. Equipped with advanced technology platforms and state-of-the-art laboratories, WACCBIP provides fertile ground for exploration and discovery.
The main impetus for the center came from some workshops that we used to do with the American Society for Cell Biology.
It was my honor and pleasure to be part of a group of cell biologists who obtained funding around 2006 for short courses on the cell biology of African diseases.
This was long before WACCBIP was even a dream, and the workshop was hosted at the old Department of Biochemistry, which exists down the road from where WACCBIP currently stands.
In 2010, with our move to this building, we had space. And also, that also coincided with a period that we had younger faculty who had undergone training in the department.
It was around that time that I had come back to the department. And the head of department at the time, who was Prof. Sami Saki, asked me to be responsible for these workshops, to be sort of the coordinator for these workshops. We were able to engage with a lot of young people in the sub-region because each workshop we
had about 20 to 25 young scientists from Ghana and other African countries. And the conversations we had and their desire to learn and the appetite for such opportunities, that was what drove us to think about how we could make this a permanent presence or a permanent opportunity for young African scientists.
I wrote a grant application designed to obtain more funds to run more of these workshops. My application was not successful, but Gordon, whose vision is much broader than mine, was able to take some of the ideas that we had put into that application and expand them tremendously and significantly into an application that ultimately was submitted to the World Bank, and many of you know was successful.
The idea about WACCBIP really was a call from the World Bank trying to help higher institutions to set up the RIM. So I called Gordon and said, Gordon, I think you should host this.
So basically, I came back to the department and I got all my colleagues together, mostly the younger ones who were full of energy and who listened to me because they were younger than me. So I put them together, Patrick, Lydia, Teresa, Osborne, Abidad. There was another lady called Marian Yako. The vice chancellor at the time, Professor Ernest Aite, was determined to turn the university into a research-intensive university.
WACCBIP first came to my attention when Professor Gordon Wandari approached me about the possibility of creating such a center. And I thought they were very important for the university. I was struck by the fact that he himself was a very young researcher. Most of the people involved with him were equally young. And I was looking for young people at the university to drive change.
An initial $8 million grant from the World Bank and further funds from the Wellcome Trust allowed WACCBIP to build a world-class center adjoining the Department of Biochemistry. Equipped with teaching labs and creating an environment that fosters scientific research and learning. The center runs master's, PhD, and postdoctoral training.
I'm very proud of WACCBIP because it has leveraged on the initial funding of $8 million from the World Bank and attracted more than $50 million for training of scientists right here in Africa.
WACCBIP's core mandate was around infectious diseases. Now, as we grew over the years, we noticed that non-communicable diseases are also coming up strongly in our part of the world. And so our funding portfolio evolved to starting to apply for grants that were involved in non-communicable diseases. Our ability to evolve with changing research interests over the years, I think, has kept us relevant.
It's been an honor for me and a privilege to help in some small way with that early development, and then to chair WACCBIP's advisory board over these last 10 years. A key success of that teaching of WACCBIP in the early years was the contribution of early career faculty, both from the north and also local faculty from Africa.
The next phase of the center was to consolidate what we had started and then to basically make an impact.
So began an investment into recruiting some of the best African scientific minds to join the center as postdoctoral fellows to conduct research and to teach.
I was looking for opportunities to come back, and because I wanted to drive research on the continent.
I was one of many immunologists at my institution in London. I'm one of only a few immunologists in Ghana. So when I think of where I can have impact, if I look back at my life when I retire one day, where can I have the greatest impact? It is in trying to build up the ecosystem back home where we are admittedly, a little bit deficient, versus making a small contribution to an ecosystem that's already well established.
As a geneticist, I was keenly aware of the depth of genetics research in African
some
people.
Fruit,
and what was striking is
coffee.
It's a huge,
especially coffee. so that we're energized for the rest of the day. We have a few changes to the schedule. So we're going to skip the plenary 6 talk, but before that, I'll call, so we'll go straight to the fellow session. So I'll call the 2 chairs who are going to chair the fellow session. Dr. Irene Owusu-Donko from Noguchi, who is also one of my favorite co-supervisors. And then Dr. Abiola Isawumi, who is also one of our fellows and a WACCBIP alumnus.
Good morning, everybody. So we'll go straight to the first fellow session. We'll invite Philip Lani, I hope I pronounced his name right, to give us his talk.
Okay, good day, everyone. My name is Philip Ilani. I'm a third-year PhD fellow here at WACCBIP, and I'm happy to share with you today some of the work I've been doing on functionally characterizing myosin motor proteins in Plasmodium falciparum. I'd like to start off this way. If I was a parasite and I was given these 2 options, to live in an island, a villa with all the luxurious things you can think of, or to live in a desert, I'd definitely go with option 1, but not Plasmodium falciparum. This parasite has chosen to reside in cells that are more or less referred to as molecular deserts. And that's because these cells lack the established protein trafficking mechanisms that are present in these other cells. And so the parasite invade red blood cells, renovate the cells to suit their purpose. And it's quite fascinating, and I think it's important for us to study the biology of this parasite. And so the problem is these parasites are developing resistance to the frontline antimalarials that we have, and developing vaccine has been a challenge over the years. And so we need to develop more arsenals to tackle malaria. If you look at the life cycle of the parasite, ranging from when the mosquito bites, every single stage requires the activity of certain key molecules. And the parasite needs this to generate force, to move molecules around in the cell, and to maintain shape, amongst others. And these stage-specific events require key proteins like the myosin motor proteins. And so looking at the erythrocyte invasion as an example, we see one of these myosin motor proteins, myosin A, providing the force necessary to penetrate the RBC. What are these myosin motor proteins? These are mechanoenzymes that hydrolyze ATP, releasing inorganic phosphate, and this way the protein is able to undergo a conformational change, and this way is able to walk along actin tracks. This process is known as the ATPase cycle, and this cycle is important for the function of the protein. Interestingly, Plasmodium myosins are quite divergent from human myosins, and that is why it gives us the avenue to be able to develop therapeutics using this approach. Interestingly, myosin A, which is one of the myosins I mentioned earlier, there's been a recent publication where a small molecule inhibitor has been identified to bind to this myosin, affecting its ATPase activity. This small molecule inhibitor is able to kill parasites in the nanomolar range. And interestingly, it can, this molecule can even kill parasites that are already resistant to the current antimalarials that we currently use. And this compound has also been shown to target several stages of the parasite development, which are desirable properties of a drug, I would say. But that's not the only myosin we have in Plasmodium. There are 5 others, but we know so much about myosin A and know very little about these ones. If you compare these other myosins, they have long tails and they have interesting domains that we really do not know what they do in the parasite, and that's been the focus of my PhD, trying to understand what these myosins are and what they do in the parasite. A paper from Rita Tewari's group tried to knock out these myosins in Plasmodium berghei, and they found that when they knock out myosin A, F, and K, it blocks the asexual development of parasites, indicating that they could be essential. And so I quickly looked at the transcriptome data available online and tried to look at the transcript abundance of these myosins. And interestingly, I found that myosin J, for instance, has an upregulated transcript abundance towards the late stage of the parasite development. And so I focused on myosin J, even though I've alongside been working on myosin F. And so the question we started asking is, are these myosins essential for parasite growth and development in Plasmodium falciparum? And to answer this question, I used CRISPR-Cas-mediated approach to generate transgenic parasites where we have added a ribozyme to the 3' end of myosin J, along with a triple HA tag. And what happens is this, once the mRNA is transcribed in these transgenic parasites, this Glimix ribozyme forms secondary hairpins, and upon addition of glucosamine to your culture media, glucosamine binds to the Glimix sequence and then leads to the degradation of the mRNA. And this way we can downregulate the protein that is produced in the process. And so I cloned these constructs into the respective plasmids, sequenced them, and once I was happy with the sequences, I went on to do transfection. When I got transfectants back, it was important for me to test whether these transfectants are really transgenic parasites or I've got wild-type parasites back. And so I used primers that are specific to wild-type and primers that can help me differentiate between wild-type and the modified locus, which I have listed in the key here. And so when I did the genotyping PCR, I was able to amplify the sequences for the wild-type, as you see in 3D7 here. And then I got successful transfectants back, although you see that I still get some wild type mixed. And so I went on to do a plaque assay to be able to select single clones of the parasite, which I then used on for the subsequent experiment. And so the next question I asked, because our protein is tagged with HA tag, I sought to test whether anti-HA antibody could detect the endogenous parasites are the correct ones. And so I started asking the question where myosin J is localized in the asexual stage parasites. And so I did immunofluorescent assays where I labeled parasites with both pre-immune and anti-MyoJ antibody, which I'll talk about shortly. We generated antibodies against the tail fragments of recombinantly expressed myosin J tail. Where you seed parasites on cover slips and then embed them in a gel, and literally you can expand the gel. Because we know the diameter of the gel we are starting with, you can actually know to what extent you've expanded the parasites. And when I did this and stained with the HA antibody, I was able to see a ring-like staining pattern in dividing schizons. And that gave us a clearer view compared to what we had for the IFAs. I just blew this up so you can appreciate it better. Although my MyoJ antibody could not give me a consistent staining, which is something I intend to go back to look at. While I was doing this paper was published, and they were able to detect myosin J in between 46 to 48 hours post-invasion at these segmenting schizons. And that kind of supports our data and even corresponds to the transcriptome data that I mentioned earlier. And so the question I then started asking is, if we remove myosin J, knock myosin J down, what happens to these segmenting schizons? Are we able to, and how would these parasites fare in growth assays? And so I induced the knockdown of myosin J by treating parasites with glucosamine, and I did not see any visible phenotype when I knocked down myosin J. I tried to hypothesize that although for some of these parasites, I saw some fuzzy-looking schizons, about 20 to 25% of them, and then I started asking the question, what is the viability of merozoites that would come from these schizons, if there's indeed an impact on segmentation? And clearly, I was unable to see any significant difference between treated and untreated when I did flow cytometry. And then I did Western blot just to show that myosin J, upon addition of glucosamine, you see the protein is knocked down. I quickly looked at the IMC to see if this would have any impact on proteins in the IMC like GAP45 and MTIP, and clearly I could not see any effect. Whether this phenotype will be the same in sexual stage parasites is something I'm currently doing in the lab. And the other part of my project is trying to look at the properties of this myosin, which I mentioned earlier. So I made this different construct, and I'm trying to produce the protein in baculoviral expression system. And these I will use for structural analysis and then try to identify the light chains, because myosins need light chains for their activity, and then also do pull downs to identify binding partners. Successfully, I've been able to make this recombinant protein in the insect cell expression system. Different fragments of it, the motor neck, the tail, and the two tails. This is the tail we use to generate the antibodies. And because these proteins are tagged with GST, when I'm going to do pull down, I would also use GST as a control. So I've been able to also make recombinant GST. Quickly, I tested if anti-His and anti-GST antibodies can detect the recombinant protein. And clearly, we see the detection here in Western blot. And even myosin J antibody, tail antibody, which I talked about earlier, can also detect the recombinant protein compared to the pre-immune, where we see nothing. To sum it up, I've been able to show you that we can generate transgenic parasites for inducible knockdown of myosin. I've also been able to show you that myosin localizes to basal ring-like compartment in dividing schizons. Upon knockdown of myosin J, we really do not see any visible phenotype in asexual stage parasites, although I'm currently looking at sexual stage parasites to see if there will be any phenotype. And myosin J deficient parasites also showed no defect in asexual growth. And I've also shown you that we can produce the recombinant proteins now, which I would use for structural and biophysical characterization. I'd like to thank all the labs that have been involved in this work. I couldn't have been able to do this without the support of these people and these really amazing mentors and supervisors for their support. I'm grateful to WACCBIP, and on this 10th anniversary, I say congratulations to WACCBIP. Thank you all for listening.
Thank you, Philip. We'll make room for 2 questions.
Well done, Philip. That was beautiful.
Thank you.
So when you knocked down MyoJ, did you check to see if any of the other myosins were upregulated, or if there was any kind of, or are you considering doing double knockouts?
That's an interesting question. I haven't done that, but yes, I intend to do that, looking at the expression of these other myosins. I would be surprised if they would have any effect, except they are maybe working together or something. But yeah, it's something I definitely would want to check.
The other thing I was curious about is how long did you do your growth assay to look for a growth defect?
That's a really good question. I did this for 48 hours. I intend to repeat these growth assays for over 4 cycles. Maybe I can sufficiently knock down myosin J over a longer cycle than just the initial one I've done. So I've done this for just 48-hour life cycle. Yeah. Divergence between humans and Plasmodium myosins. I find these proteins very interesting because, to me, they seem to be all over the place in the parasites, and there are about 6 of them. We know so much about just one, and so I decided to look at the others, what they do. Interestingly, back in 2022, 2023, the paper on myosin A came out where they identified an inhibitor, KNX002, which binds to myosin A, reduces its ATPase activity, can kill parasites, at different stages of parasite development, can even kill parasites that are resistant to the frontline antimalarials that we have. So I thought it would be an interesting shot to look at the other mycins. And mycin A, the inhibitor that is targeting that mycin, there's a whole company in the US now.
Hello, Eunice, can you hear me? Eunice, if you can hear me, can you put your presentation in, your slide in a presentation mode so that we can start the presentation? Please, technical group, can you hear Eunice? Can you confirm if she can hear me?
Yes, I can hear you.
Okay, please go ahead with your presentation. You have 12 minutes.
Thank you very much. Good afternoon, everyone. My name is Eunice Ampezia Iraq. I'm a second-year PhD fellow, and I'm going to give a talk on the Pacific Island and genomic structure of Klebsiella obtusa indicated in mucosal infections in Ghana. So Klebsiella species are important human pathogens that form part of the normal
All right, thank you, Eunice, for the excellent talk. We have time for 2 questions. Okay.
Thank you very much for a very interesting talk. I wanted to know about the lava in vivo infection model, and if it's relevant only for Klebsiella or other pathogens as well.
You can use it for other species, not just Klebsiella.
Interesting, thank you.
So do we have any other question?
Thank you. Thanks for a very interesting, very nice talk. Could you tell us a bit more about the epidemiology or the public health perspective on this pathogen? Is it a widespread problem in different hospitals? Yes, thank you. Yes, please.
All right, thank you, Eunice, for the talk.
So we'll take the final talk from Harry Hayford.
Hello. Can you hear me? Okay, good morning. My name is Harry Hayford, and I'll be presenting on the role of histamine production in Klebsiella pneumoniae antimicrobial resistance and virulence. So histamine, as you already know, plays major important roles in the human body in neurotransmission and pro-inflammatory responses. However, exogenous histamine from other sources outside of the human body have been found to cause allergic reactions and cause histamine intolerance. The major sources of these exogenous histamine is as a result of the decarboxylation of the amino acid histidine to produce histamine in bacteria. These bacteria produce histamine in order to survive extreme acidic pH so that they can survive in these acidic pHs. The most common bacteria, or one of the most common bacteria that produce histamine in large quantities is Klebsiella pneumoniae. The bacterium is a gram-negative bacterium that has been characterized as a global priority pathogen by the World Health Organization and may cause different complications based on the area of localization in the body. It is currently treated with cephalosporins, quinolones, and carbapenems. However, resistance to these antibiotics, as well as other classes of antibiotics, is a common occurrence in Klebsiella pneumoniae, which is thought to be spread through the presence of high-rate clones and resistant plasmids. In addition to this, the bacterium can produce bioforms which enable them to survive in the environment to cause nosocomial infections And also, they are able to produce capsules which ensure their survival against phagocytosis. So although histamine production has been characterized as a survival mechanism to environmental stress like acidic pH, the effect of histamine production on other survival mechanisms like antimicrobial resistance and biofilm formation is not known. So this study sought to investigate histamine production in Klebsiella pneumoniae by detecting and quantifying histamine production in vitro, determining the effect of histamine production on antimicrobial resistance and virulence, and also determining the effect of histamine production on the expression of antimicrobial resistance and virulence genes in Klebsiella pneumoniae. So the study employed 4 strains which were archived. 2 were clinical from septicemia patients and 2 were environmental from hospital formites. For the histamine detection assays, we use E. coli EK19 as a control. So the experimental approach was basically first to detect histamine, quantify it using enzymatic methods in HPLC. Next, we did comparative antibiotic susceptibility testing by determining or testing the resistance and susceptibility of the histamine-producing and non-histamine-producing strains of Klebsiella pneumoniae. Then we went ahead to do infection assays to determine the effect of histamine production on virulence in Klebsiella pneumoniae, and also expression profiling using RT-qPCR to determine the effect of histamine production on the expression of resistance and virulence genes. So to begin with, we found that the strains could produce histamine. And this assay is the Nevin's calorimetric assay. The change in coloration of the media from the brick red coloration to the purple coloration, as can be seen on the screen, is as a result of histamine being produced. So the media is acidic. Once histamine is produced, it changes the coloration of the media to purple, which is as a result of the increase in the pH of the media. We determined that histamine production in all the strains began at the log phase. We did this by isolating cells from both the log and stationary phases and testing them for histamine production. In this case, we found that since we found that histamine production was produced at the log phase or began at the log phase, it made us hypothesize that because it is required to survive the extreme acidic environment, then there is the need for the strains to produce histamine even before the log phase in order to undergo exponential growth. Next, we used HPLC to confirm and quantify the amount of histamine produced. In all the tests for the strains, we found that we detected histamine at a retention time of 26.5 minutes. And also, the strains could produce histamine between 390 to 800 parts per million. In all strains, we found more histamine was produced at the stationary phase as compared to the log phase. This could be indicating that histamine could be advantageous in the stationary phase in order to survive the increased bacterial competition, which might be due to the production of antimicrobial secondary metabolites. Next, we screened the strains for the presence of the histidine decarboxylase gene, which is responsible for histamine production. We found that in all the strains, the gene was present. So going forward, we wanted to determine the effect of histamine production on antimicrobial resistance and virulence. So here, what we did was we induced histamine production in the strains and then just did antibiotic susceptibility testing and all the other tests for the phenotypes. So after ASTs, we found that the resistance profiles of the strains did not change after histamine production, indicating that the strains were still resistant to or susceptible to the antibiotics they were resistant to before histamine production. However, we found that there was increased antimicrobial resistance levels after histamine production. So if you look on the screen, the previous MICs, that is the minimum inventory concentrations that could reduce the bacterial growth to less than 10% before histamine production, were no longer effective in reducing the bacterial growth to less than 10% after histamine production, as is shown on the screen. And after doubling the concentrations, that is the minimum inventory concentrations, we found that still they were ineffective in reducing the bacterial growth to less than 10%. In the case of the antibiotic meropenem, we found that after histamine production, when you increase the drug pressure by doubling the minimum inventory concentration, there's actually an increase in survival, meaning that there's increase in resistance after histamine production. Next, we determined the effect on biofilm formation. So here we observed two things. In the case of strains that produce high concentrations of histamine, here, you found that after histamine production, there is reduced biofilm formation. But in the case of strains that produce lower concentrations of histamine, after histamine production, there is increased biofilm formation. So this is indicating that histamine production is reducing the need for biofilms to be produced. So biofilms are also produced in bacteria to also help them to survive their acidic environment. So in the case where there's more histamine being produced, there's lower acidity in the environment. That means the environment is more favorable for the bacterium to survive. So it requires it to produce less biofilms in order for it to survive. Next, we determined the effect of histamine production on encapsulation. We found that after histamine production, there were more encapsulated cells as compared to before histamine production. This could be indicating that histamine production could be driving the cells to survive phagocytosis. So to investigate this, we infected human macrophage cell lines with both histamine-producing and non-histamine-producing variants of Klebsiella pneumonia. And then we found that histamine production did not cause a difference or a change in the viability of the macrophage cell lines. That is, they still had the same degree of cytocidal effect on the cell lines before and after histamine production. However, we found that after histamine production, more cells could survive the macrophage phagocytosis by the macrophage cell lines as compared to before histamine production. So this can be attributed to two things, may be attributed to two things. It's either from encapsulation of the increased encapsulation in the cane pneumonia strains, or it could be as a result of the effect of the histamine itself being produced on the macrophage cell lines. So next, we screen the strains for common resistance and virulence genes that are normally observed in Klebsiella pneumonia. We found that the strains harbored at least 2 resistant genes for beta-lactams and efflux pump genes. In the case of virulence genes, the strains harbored all the strains harbored virulence genes for fimbriae, siderophos, and membrane lipoproteins. So to determine the effect on gene expression, we use RT-qPCR. We found that in the case of the resistant genes, they were downregulated after histamine production, whilst in the case of virulence genes, the virulence genes were upregulated by histamine production. Now, the upregulation of the virulence genes was expected because there was increased encapsulation after histamine production, and also in the upregulation of the fimbriae genes could be indicating why biofilm formation was altered after histamine production. So, in summary, we found that histamine production increases antimicrobial resistance levels in Klebsiella pneumonia. It complements biofilm formation for survival in an acidic environment, and it increases virulence by promoting survival in macrophages. And since the strains could produce such high concentrations of histamine, it indicates that they have the possibility to cause histamine intolerance in addition to all the complications they might cause. So in the presence of these evidence, we could say that histamine production is a significant public health threat that should be investigated further. I'd like to acknowledge my supervisors, my labs, and the Ghana Standards Authority for the study, and also WACCBIP for funding. Thank you.
Thank you, Harry. We'll take 2 questions.
Those who are more susceptible to allergens,
yes,
are more susceptible to infectious diseases. Okay, so the studies have shown that for some kinds of diseases, so for those who are more susceptible to allergens, they have larger populations or higher populations of bacteria that harbor the HDC gene. So in the case of asthmatics, and also people with irritable bowel syndrome, studies have shown that they have larger populations of these bacteria in there. But as to whether they are more susceptible to all infectious diseases, I can't really talk about it.
Thank you. I just have, I think it's more of clarification for me. When you did you said histamine increases the phagocytosis of the Klebsiella in macrophages.
It promoted their survival in the macrophage cell.
So they make the cells survive?
Yes.
And then so the cells survive, the parasite survives?
Yes.
The bacteria survive. Okay, thank you.
No more questions. All right, thank you very much, Harry.
All right, thank you, Harry. There is a slight adjustment in our program. Earlier today, we missed a plenary talk by our very dear own Dr. Yao Aniwe. So we'll be having Dr. Yao now. He's a senior research fellow here at WACCBIP, at the same time, the head of technology. Dr. Yao Aniwe, you are welcome for your plenary talk, please.
No problem.
Okay, thank you very much for the opportunity to present what we've been trying to do. Because I've already taken your time, I will not spend much time. I will present what. So, looking at malaria, there are a number of parasites that are able to cause malaria. Like the presentations that have come before, you will hear Falciparum as the major. But notwithstanding, there are other parasites that are also driving malaria in our settings. So in Ghana, we have malaria in ovale as one of the major. And you will see most of the time, anytime you have Falciparum, the major culprit who is underlining also, or going where it is Plasmodium malariae and Plasmodium ovale. Most of these are known to cause benign disease. That is what we have known them for. Others, chronic infections of P. malariae has been associated with nephrotic syndrome. Other complications have been shown. Recently, there are a number of reports of anemia associated with monoinfections of these parasites. So the simple question anyone will ask is that, really, is it the case? If you look into our population, if chronic infections of P. malariae can drive nephrotic syndrome, then could it be that a number of people who are in the kidney clinics, we should begin to ask some of these questions in there? So what we have been trying to do is to help us be able to, first of all, detect them, and then we can now begin to look into different populations. One thing which is very common with these parasites is that they always occur in low parasitemia, and most of the time occur in co-infections. One thing is the diagnostic tools we have. Whether microscopy, you need a trained microscopist who is diligent enough to find these. Partly also, the RDTs we have don't pick. Most of them are HRP2 based and therefore wouldn't be able to pick this except pan-LDH. So that is also another question. Since the pan-RDTs are more expensive, hospitals don't use them mostly. So if you look through literature, you'll find that there's not much done on these other parasites, partly because we consider them neglected. So recently there have been reports that after treatment with ACTs, these parasites persist. So the simple question that everyone will ask is that, do the ACTs actually have direct impact on non-falciparum malaria? And recently, with growing data, clearly we are seeing that it's not that effective against the non-falciparum malaria. So if you to look at falciparum in the blood, you will always find the ring stages. But if you get to P. malariae and P. ovale, you could find all the different stages, including gametocyte in the blood. So this is one of the unique challenges also with them. With P. malariae, the life cycle is a 72-hour life cycle, while these are 48-hour life cycles. P. malariae is known that it is able to form a hypnozoid, meaning that it forms a dormant form which hides in the liver, and whenever they are happy, they will come out. So if you give somebody a drug and then the drug pressure wanes, they decide to stay or decide to come out. So these are the conundrum. But notwithstanding, recently there have not been any data really pointing to the hypnozoid forming ability of P. ovale, which is still something to be explored in the community. So one particular thing in the lab, we developed tools, courtesy talented PhD students and graduate students. So these allow us to be able to now detect the non-falciparum which occur in low parasitemias, now with good confidence, and then we could go and classify them. So one particular thing is that clearly, if you look in this, what you see is that falciparum, this is a clinical case. We always have falciparum being the dominant, which is what we know. But notwithstanding, you could have up to 20% of the people going to the hospital being infected with malaria as well as ovale. Then when you want to segregate this and ask the simple question, do they occur as monoinfections or mixed infections? You'll find still you have monoinfections of falciparum dominating, but you have mixed infections of falciparum and malaria being the second, and then you have monoinfection. And in some individuals, we could have somebody having all the three jubilating in the person. Yeah. So in terms of age distribution, this one, we need not belabor it. It occurs, it's very common in the young, so you can see that. But notwithstanding, still, in these individuals, you will find malaria. With the age group distribution, you find the same pattern. So clearly, here is just to show you that if you use microscopy and then the qPCR, we know qPCR is at a molecular level and therefore it really helps. With falciparum, because we always have higher parasite numbers, it is easy to find them. But with that will have broad spectrum, and as you will have listened to Franklin's talk yesterday, we're trying to look at the CSP, which is cross-reactive to falciparum, ovale, and malaria. Yeah. So here specifically also you will see that those who have mixed infections, yeah, there are not too many, but actually they are there. And we have to be concerned and to see how interventions targeting these individuals will help us drive the burden of the disease down. Yeah. So one particular thing that we try to do is we have these parasites. They are co-occurring. We have fewer monoinfections. The simple question is that, can we now develop tools to help us be able to study their drug susceptibility pattern? So what we did was to get monoinfections of P. ovale and P. malaria. And the other criteria is to be able to see individuals who have relatively higher parasitemia, and the composition of the different stages of the parasite should be biased towards ring stages so that you can now follow them through. So with this, we developed the protocol for P. malaria. And here I show you susceptibility. We compared the protocol between Ghana and Mali. So here, using chloroquine, you will realize that, yes, most of the parasites in the population have become susceptible to chloroquine, but with P. malaria, you still get a few that shows kind of resistance. Comparing it to quinine, others, if you look at Artemita, there is this parasite that decided not to really succumb to. One particular thing is that there is the likelihood that some of these parasites are more of the late stages. But if it is the late stages also, we expect these drugs, which we know kill trophozoite and schizon stages very well, to be very effective in killing. But then, irrespective of whatever stage, we still get some of the parasite that shows a higher level of tolerance to the drug that is used here. So the other thing is that we did this same study also. The earlier data is Mali, here is for Ghana. We find some malaria that is also. So this is actually the simple question we're asking. Are these parasites resistant? And if they are, what are the unique markers? As we can say now, most of the resistance we know, and the associated genes are all for falciparum. We don't know for malaria and ovale. So this is one of the things that we're doing to now begin to go down and ask these questions. What genes are driving this phenotype?
A very beautiful talk, by the way. We'll take questions, but I would like to start. So, was there a reason why you picked Mali Was there any specific reason comparing Ghana and Mali?
No, we could have used any other, but I think with Ghana, Mali, I have an active collaboration there, and this protocol was developed in such a way that both labs could test. So now we're bringing in Benin and other countries to equally adapt to that.
All right, thank you. Yeah. Okay, we have a question at the back. So there's a question.
Yeah.
Can I?
Go ahead.
Yes. Go ahead.
Okay.
Please.
Thank you very much. Thank you, anyway, to present this. Okay, I'm very concerned about what you've said. You said Plasmodium ovale and malaria are really very low in terms of prevalence. But at the same time, it can happen in symptomatic and asymptomatic. So my question is, did you actually test this in hospitals? And in that case, don't you think there has been a selection bias and showing that this prevalence might be high, much more higher than what we think?
Yeah. Actually, we actively telling people to be testing for this simply because, one, there is a hypothesis that when you treat, because they are less susceptible to the ACTs, they will be amplified. So there's a recent paper from Tanzania showing a community that to them, they've driven falciparum down and they are seeing malaria come up. So that is one hypothesis. The other thing is what we call the comparative inhibition. In the presence of falciparum, the others are quiet. And as long as falciparum decide to go with any drug or any intervention, then they now get up to jubilate in the house. So these are some of the things. There are not many people testing. Because most of the testing we do are PCR based. That cannot be done in the hospital. But gradually, with tools like LAMP, we are getting the hospitals to also begin to test this. Yep.
Okay.
Yes, thanks, Theo, for that nice presentation. Just to check, for the complexity of infection, because you said you detected more monoclonal infections, but I think in some other publications, including ours and yours, there seems to be quite a lot of diversity in, which is not geographically restricted, in P. malaria, for example. So it was quite surprising that having that level of complexity population-wise, single infections were always monoclonal. Do you have any explanation for that? The second is the work that you're doing across laboratories. For P. falciparum, we found out that its variability between laboratories, even though you're sharing protocol, can be a significant nightmare, a significant nuance to the data, and to be able to compare data between labs. So what are you using to normalize the phenotypes you're getting, IC50s in this case, between the two labs? Because it will become quite difficult to compare if you don't have a comparator as a control that you can reference to.
I think the last part, I don't know, did you guys hear?
Yeah, the last one was how do you compare? Because you showed those dot plots comparing Mali and Ghana.
Yes.
And my problem there, as for P. falciparum, is that you cannot really compare assays between two labs because interlab variability is quite high. Even within the same lab, two individuals running two assays for the same isolate for IC50 may end up giving you different results in terms of numbers. So the distribution of those numbers compared between two labs, how do you control and normalize between the two?
Okay. So with that, what we did was we did the assay. We initially used falciparum. So the 3D7, DD2, and K1 are the parasite we use. We did the assay in Ghana. We have the IC50. But also with the Mali, I had to go there, help them set up so we could do same and make sure that the level of reproducibility in there is comparable. It wouldn't be the same, but it's comparable. Yeah. The other thing with the complexity of infection in terms of the different species, one thing I think is, So a work done by Felix in the lab, we took, say, Central Region specifically. We moved from town to town, and maybe town A, you'll find Falciparum and Malare. You go to the nearby town, you don't find Malare, you rather find Ovale. So the question has to also do with, is the vector competence the same? That is the other thing that we don't know. And I know there are groups who are looking into that. The other thing is also, in terms of the multiplicity of infection having the different species, what I know, because all these people or these individuals, we check their hematological indices, what is typical known to correlate with, say, like platelet count, the hemoglobin levels, are the same parameters that flag for all these as well. So, I mean, this is actually the reasons why we're amplifying this, given this talk is equally amplifying, to make sure that we all become active in looking for them and also looking towards finding interventions that will help us clear them. Yeah.
I think because of time, okay, we can have 2 more questions, then we can continue the conversation over lunch. So we can have, and then we'll come here, please.
Okay, so with respect to the mixed species infections, did you look for gametocytes so we can understand the level of competition between the species to get transmitted to the mosquito stage? So if you have Falciparum and Malare, which one is making more gametocytes? Yeah.
So if you look at individuals that have mixed infections, you will mostly find, especially Malare, you'll find the Malare gametocyte. But that, I wouldn't compare it immediately with Falciparum of the same individual, because Falciparum getting into gametocytogenesis is triggered by a lot of factors. But with Malare, we know that, I don't know the biology driving it, but at any particular point in time, you sample somebody who has Malare, you find ring, you find trough, you find the gametocytes. This is one thing, but I don't know whether is it that they cannot sequester, or possibly there are unique mechanism they feel, let everyone go out and play, and if one is killed, the others will survive. I mean, these are all hypotheses that are to be tested. Yeah.
Okay.
Thanks, Jiao. So many years ago, when there was high Falciparum transmission on the coast of Kenya, we looked at barley phenotypically, not using PCR, and found that although there were very low apparent prevalence, in fact, there was a very sharp age drop-off in the very young, implying that, in fact, probably it's being transmitted very widely, and then becoming immune quite quickly. I wonder whether you see the same pattern. And also, I think, to simplify it, you talked about Avali rather than the 2 species of Avali. Do you see any difference in distribution of Curtis eye and Walker eye?
Yeah. So this is one thing that specifically I was about 4 or 5 cases were referred. Somebody who had been given antimalarials and still went back, and the person came when we checked. It was Ovale Walikari. So kind of, in a way, we see more of Walikari more associated with mono infections getting to hospital from our hands. This is what we're seeing. And also, Ketisi, you get them mostly with the data we have. All the samples we get from the community, the asymptomatics, we do find Ketisi as well more. Yeah.
All right, thank you, Dr. Aniwe, and again to all our presenters for this session.
All right, let's thank the chairs for a great job, well done. And as we say, time flies when you're having fun, right? It's almost time for lunch. But before we go for lunch, just a quick note about the poster sessions. So we will have turbo talks today, and the people who have been pre-selected to give turbo talks, the odd-numbered abstracts will give their turbo talks today in chronological sequence, so 1, 3, 5, and so on. So you have to make sure, even though we asked to submit the posters for us to print, those that didn't print, you have to make sure that you put up your posters if you're an odd-numbered poster, so that people can go around and look at it today and judge it during the poster sessions. So that's it. And for your poster numbers, look in the abstract booklet, which is online, if you haven't already done so. So without much ado, we're going to break. I think it's early. Yes. We're going to break early for lunch, which will give all of us a chance to walk around, look at the booths, speak to the vendors, look at the posters, speak to the presenters, and we'll be back here at 2 p.m. sharp for the keynotes from Professor Basiru Bonfo. Thanks for being sharp, and thanks to all the speakers for keeping within time.